So first, a couple of things to say about me. I'm actually coming from the mathematical modeling point of view. So machine learning is not something that that is my expertise or anything, but I think that a mathematical modeler has to use the tools that are needed, not the tools that one knows. And in this particular case, machine learning comes quite handy. And the second thing is that my academic name is Perez Velázquez, so most of my, actually most of my publications are on this name, so not on Judith Cerit. Yeah, so this workshop is actually about the hallmarks of cancer, but they are present once the disease is actually already developed. And the question is, are there some things that precede these type of hallmarks? Are there indicators that will eventually go on and develop into these full-blown hallmarks traits that we all know that we discussed in the early session? So there are actually some indicators depending on the type of cancer that comes into question, and these early indicators could have different could mean different things. They could, for example, be molecular changes. They could be cellular changes. They could even be only symptoms, and they are important because they, in principle, offer an opportunity to act early, and they basically could potentially buy us some time to react. So this is the part I want to concentrate. We could, in principle, try to correlate these early indicators with the hallmarks of cancer. So some of these, as I said, some of these early indicators will eventually go on into establishing what now what later will be the more recognized hallmarks of cancer. For example, if the changes in these early indicators are genetic mutations, eventually they will be recognized as one of the known hallmarks, for example, resisting cell death. If this early indicator happen to be, for example, chronic inflammation, they eventually will, if the disease progresses, be present as, for example, inducing angiogenesis and so on. So all these early indicators, as I said, will, if the disease progresses, lead us to recognize them as hallmarks of cancer. And what I want to concentrate on, what are the opportunities we have to detect this early enough. So there are specific methods currently, depending on what type of early indicators we are talking about. If some of them are, for example, mutations, then there are genetic testing for these mutations. If those changes are epigenetic, then we could test for example, abnormal DNA methylation patterns. For example, there are also for some particular cancer, there are inflammation biomarkers. There are also imaging techniques, such as mammograms, for example, in the context of breast cancer. And there are also some tests like liquid biopsies. We could also monitor the immune system. So these methods will be what they become the data that will feed a database approach. So that's what machine learning models are. They basically use data to try to answer different questions from the data. And as we know, they have been proved to be very helpful in several stages of cancer diagnosis. They are capable in many cases to go above what human detection capabilities are. They are, in some cases, they outperform pathologies, and this all, of course, help us because they, in principle, could help us to detect the disease very early. There are, in fact, already some FDA-approved tools for cancer diagnosis. These are some of them. They have been for a couple of years already approved, and they work for specific cancers, and they have proven to be quite useful. Let's remember that machine learning has a specific workflow that needs to be followed. This is just an example for an image-based example where the data that comes into questions is data from the patient, for example, image from pathology, and this image, they become the data that are going to feed the machine learning models. And typically, one still has to pre-process the data. If, for example, you have an image, we will produce several tiles out of one particular image. Then we have to choose the machine learning model. Very typically, one model is not enough. We may need to combine it with other models. And what we normally do in this particular case is come up with some sort of prediction, typically in the form of a probability. However, as I explained before, I don't come from this area. I'm a mathematical modeler, and these type of models have received several criticism, and one of them has been consistently the fact that these models, machine learning models, tend to be black boxes. And it's sometimes difficult to know why the model is doing what they are doing. And it's one of the main critiques that I have from this type of models. Very consistently, explainability is one of the main challenges that these type of models offer. Particularly when the model has already produced some sort of output. This is from a review we did a couple of years ago, where in computational pathology, typically when the model is already been chosen and deployed, when it comes to interact with the pathologies, it's very difficult to explain why the model is doing what it's doing. So this is the point that I want to address, the explainability side of things. Is it at all possible to achieve that? In general, it's very important because if we want to use them, machine learning models in clinical practice, the models need to be explainable because otherwise we cannot track down the reasoning that is leading to a particular suggestion. They are actually necessary for compliance and trust. And of course, it has the benefits that in order for the model to be successful and aid the medical professionals, they need to be explainable. And in the future, if these models are explainable, that will increase trust in this type of models, and this type of models will become more widely used. So how do we how does explainability come into this type of models? The problem with machine learning models is that they don't typically directly find relationship as a cause-effect relationship. What they do is they link the inputs that they receive with the outputs based on a particular pattern. And what we want to do is trying to understand what happened during the process. So there are basically two ways to bring explainability into machine learning models. One will be after the model has been trained. There are specific methods that one could do with for example, with a neural network that has already been implemented, and try to bring the explainability after the training, or we could actually build the explainability part already into the model. So there are these two ways to go about explainability. Obviously, the models with the explainability already built in are more reliable for explanations. But this normally is not this is not always possible. So I wanted to show you an example that I really like that actually is it describes really well that this is in fact possible to do. This is an example of early gastric. This is an example for gastric cancer, and the type of data they used was DNA methylation analysis, and they basically analyze DNA methylation patterns, and they actually achieve very high accuracy with this model. And through this process, they were able to identify novel epigenetic markers, because what they did was they tried to rank what are the features that are more likely to play a role in the development of this particular disease. And as a nice outcome from the model was they, by detecting these biomarkers, they at the same time implement screening protocols. Let's have a look at what they did. So they were able to rank different features associated. This is early indicators, as I mentioned before. So those are all those are typically early indicators of gastric cancer. They were able to rank which of these are the most related to will go on to develop the disease. They came up with the fact that if the individual at some point presented gastric disease, that already places them in a with a very high chance of go on to develop gastric cancer. The second one was night sweats. Not that close in comparison with the first one, but by being able to rank explicitly all these different features, we can clearly and explain in a explainable manner see what happened with the algorithm. How did they do that? What are the mathematics behind producing this type of ranking? This is normally done in this particular case through what the so-called weighted assemble models. They are assemble models because they are an assemble of different models. And what they do is they basically, one considers the weighted sum of the outputs of different models. So we have model 1 that has one particular ranking of these features, and model 2 also ranks these features as well, model 3 and so on. And then at the end, we take a weighted sum of all these of all these feature importance, and with that we come up with one single one. Typically, is in the picture is not so clear, but typically the type of model that is used here are called decision trees. Decision trees are a method which is very commonly used because it provides a visual representation of what is happening. And we could think of decision trees as asking a series of questions. So, for example, did the patient have gastric disease? If that happens, then the algorithm puts them in a particular group. And then another question is asked, did the patient did the patient have genetic history of this particular disease, and so on? So a series of questions are asked until we end up with one particular group. And as I say, they happen to be very useful because they are simple. It offers a visual representation. That you want to go back and see what happened, the algorithm will show us. We could produce this image. They're very easy to implement, surprisingly, and it explains what happens during the process. Another example, this is in this case for breast cancer. What they did is the type of data they used were mammograms, and the explainability part came in two parts where they were looking for feature extraction to see in the mammograms, we could see particular patterns, for example, the texture of the cells, the structure of the cells, particular areas within the image. And they came up with a particular method that they use called asymmetry analysis, where they realized that asymmetry between the mammograms of the right and left breast, if they actually happen to be very asymmetric, that we actually have that happen to be a significant marker for breast cancer risk. And it has clinical relevance because one could straight away give an explanation, a suggestion to the clinical professional. And they did this for with an associated probability of developing the disease as early as 5 years before the presence of the disease. And as I say, what they developed was this comparison between the right and left. Mathematically, what that means is called bilateral dissimilarity. And the mathematics that are behind that is it basically divides the image into regions. And every region is represented by particular features, for example, the pixel intensity or the shape of the region. Then distance is defined between the regions, so how different these regions are. It could be a very basic distance or even more complicated distances coming to question here. And then one has to normalize this and set up a particular scale to be able to compare, and then aggregate everything to come up with a calculation to rank them all. And through this process, then we can actually select for particular features. For example, that will allow us to identify biomarkers. In the example that I gave will be the difference, the fact that there are differences between the right and left that actually happen to be a biomarker. There are several techniques involved, it has a lot of applications because it could be used for other types of cancers, for other types of contexts as well. And yeah, I'm running out of time. Yeah, so I only focus on what one could do, what one could do within the diagnosis part. But actually, machine learning could be used in other parts. For example, once we have gone to the diagnosis, we could, for example, think of using this type of models for prognosis or even for treatment and monitoring. I believe that the future of cancer research and cancer treatment will require an important advance of the use of machine learning algorithms. It's very important that these are explainable in order for the clinical professional to use them. So the research has to be parallelly developed in this area. And from my side, I just wanted to mention what I had in mind. We have planned a collaboration with Abraham Silva, my collaborator who works for a pharmaceutical company. And here we want to look at pre-neoplastic lesions in papillomavirus. In this particular case, these early features that I was talking about may include these neoplastic lesions. So they happen before seeing any neoplastic tissue. And these are two examples of them. And the related hallmarks will be evading growth suppressors or resisting cell death. And what is the type of data that comes into question here? There will be pap smear tests and HPV testing. A pap stain is actually it's ultimately an image, and it's basically a microscopic image of the stained cervical cells, and we could use this as basis. In this case, the machine learning models can be trained with this image, and what we'll be looking here will be things like cell size, shape, nuclear to cytoplasmic ratio, texture, other patterns that may be already known by pathologists, but may be detected as key indicators. Yeah, so that was from my side. As takeaways, we could say that it's very important. This issue of interpretability is very important because it ensures transparency and trust. The part of the early diagnosis is the most important thing to mention, because this is where these early these predecessors of the hallmarks come into question, and it has a lot of application. I just showed you the two examples. Of course, the use of this type of model represents several challenges. For example, the fact that data with a good quality has to be available. Yeah, sometimes this interpretability side of the model is not so easy to implement in the models. For some particular scenarios, may not be so easy to integrate into clinical workflows. And yeah, that was from my side. So I look forward if anybody has questions or comments.